Hello. Good morning. Thank you for coming. Thank you very much for the introduction. And my name is Wang. I'm a postdoc here, and my PI are Dr. Jason White and Dr. Emmer. So the title today that I presented is I'm presenting as Nanomaterials in Plant Nutrient Enhancement, Disease Suppression. And okay, and agrochemical delivery. So first of all, I would like to start with some background of this project. So as we know that although the green revolution has led to significant increase in the agricultural production, but currently many of the processes are not sustainable. And the efficiency of the agrochemical delivery is quite low. So we need to find a way to increase it. The agricultural production will have to increase by up to 70% because of the dramatic increase of the global population. And also, as we know that the changing of the climate may negatively impact the yield of the agricultural crops. Besides that, the plant pathogens also can reduce the yield of most crops. And here, that what I'm started is the fusarium with tomato plants, as the picture showing here. And although recent work has demonstrated the unique potential of the metal-based nanoscale materials to enhance the tolerance to this disease, however, the effect of those metal-based nanomaterials are controversial. Many other literatures also show that they may have negative impact to the crops. So how to solve these problems? Here I would like to introduce the elemental sulfur. This is a secondary nutrient. It's a key element for the production of formation of essential amino acids in the plants. It also related in several very significant metabolite biosynthesis pathway, like within the defense system against the biotic and other stresses. Besides that, from this literature that show the change of the ratio of the pathogen is highly related to the concentration of SO2 in the air, which means that sulfur may play an important role in the disease suppression of the plants. And interestingly, nowadays that since the use of high sulfur contained fossil fuels has decreased, so the input of sulfur into the soil are decreased. So right now, people are actually trying to fix this problem by increase the sulfur content in the soil. Thus, the application of bulk sulfur has become more common. So if you like Google the sulfur for the plants as a fungicide, you could find a lot of commercially available productions. However, this still leads a problem that the inefficient delivery of those bulk sulfur can lead to soil acidification. In this graph, it shows the mechanism of how it happens. So if you apply the elemental bulk sulfur into the soil, it will transform to this SO4, and if this sulfate is lost by leaching, then the soil gonna result in like more acid than before. So I think for us to solve this problem, we have like two steps. or two methods. The first is we need to find a way to reduce the excessive application of elemental sulfur into the soil, but without decrease the function of it. And the second thing is that can we find another pathway, like instead of transform to this sulfate, can we find another pathway that we can deliver those elemental sulfur into the plants, but without the generation of additional hydrogens? So these are our the two aims of our studies. The solution for right now that we found is to use the sulfur nanoparticles. So as we all know that here this very famous talk, there's plenty of room at the bottom. So it seems that from 1959, after this talk, people started to be interested in the nanotechnology. And as we all know, the nano is not just the size. So this is a very a good example. So if we decrease the size of a bug, like any item, for example, this is my cat. So after we keep decreasing the size, instead of getting a smaller and smaller cat, and in the end, when it reach nano size, we will finally have a dog. That means after the size change, the property of the original item can be changed dramatically. Another example is here that by changing the size of the gold, which we normally know as the color should be golden, that all this color belongs to gold, just because of the size-specific effect. Another good example is this here, that this is an ancient Roman treasure from about AD 400 years. And this glass gonna change color from originally green to the red, just because when the light comes through. This is because this treasure, this cup, is made by gold silver nanoparticles by our ancient. So so I would say the nano size give us a lot of possibility. So in this study, the material that we're gonna use to as an alternation to the bulk sulfur is the sulfur nanoparticles. And another reason is that it has been demonstrated that the nanotechnology has been demonstrated to be very promising in the agriculture field. There has been significant interest in using nanotechnology in agriculture to increase the crop yield and also the qualities. So if we search the nanoparticles and plants, here are the results that shows how this interest grow from 2011. There are about more than 1,000 paper to last year that there are more than 1,500 paper related. And there are many other literatures shows that by using the nanotechnology, basically the nanomaterials, that we achieve a lot of increase in the crop yields and also the disease suppression. However, the mechanism behind is still not clear, so that leaves us plenty of research questions. And the first question for us is how to apply this nanomaterials. So we have already decided that we're gonna use, the hypothesis is to use nanosulfur to to suppress the Fusarium disease in tomato plants, and how to we need to find a way to apply them appropriately. So so first of all, that we know that the Fusarium is a soil-borne pathogen, and in this graph it shows how it affects the plants. It starts from the interaction with the plant root, and once inside the root cortex, it gonna translocated upwards through the xylix to the shoots. From one of my previous study that I proved that the nanoparticle could be uptaken by the plant root via the xylem pathway. So the way that we apply those nanomaterials is the soil application. And from another my previous study that we found after uptaken by the roots, the nanomaterials could be translocated from root to shoot and aggregate in the leaf tissues, like differently depending on the particle size and also the plant species. So, for example, in these bug-tray leaves, that the nano-sized copper oxide and bug-sized copper oxide are differently distributed. This has been proved by the two-photo microscope images. And by the ICP-OES, we demonstrate that we further demonstrate and quantify the translocation of those copper-based materials that we applied in the soil. So so based on this, that we're gonna apply those sulfur material in the root, and after they've been uptaken by the plant root, they're gonna translocate it to the shoot and help to suppress the soil-borne Fusarium disease. Another question about the application way is the concentration, surface functionalization, and charge of those nanoparticles. Again, from some of my experience, some of my previous literature, that we demonstrate that both the concentration, the surface functional groups, and also the charge plays an important role in the uptake and interaction of those nanoparticles with plants. So here, for example, I applied 3 different types of nano-TiO2 to character plants, and one is the Pristine with pure nano-TiO2 with a negatively charged, and the other 2 are surface coated with different functionized groups, which is one is hydrophilic and the other is hydrophobic. Both of them have a positively charged. So because of this, we observe different responses from the character plants to the 3 different types of nano-TiO2. So which means that when we apply the nanomaterial, we need to take care of the concentration and the surface properties. So based on all this, we design our greenhouse experiment. So the materials that we used is the Pristine nanosulfur, a surface coated nanosulfur. And to compare the size effect, we also include bulk sulfur and ionic sulfur. The concentration we choose is 0, 100, and 200 milligram per liter soil based on previous studies and literatures. For for the treatment, we have infested and non-infested plants. So here a picture shows how we prepare the material and the treated soil before to start the greenhouse studies. And we actually did 2 greenhouse experiments. In this one, we use similar design, but we for this one we want to study the mechanism of the interaction. So we designed a time-dependent harvest. We harvest the tomato seedling at 4, 8, 16 days after the transplanting to the fusarium infested soil. Here, I'd like to show some characterization of our sulfur materials. So this is the first. This one is the SMEDS that shows. This is the Pristine. The non-coated nanosulfur. It shows only sulfur, and the next one is the coated nanosulfur. Sorry, I forgot to mention that the coating is stearic acid. This coating is helped to firstly modify the surface, like the zeta potential of the nanoparticle, and also prevent the aggregation of those sulfur materials. So from this EDS, instead, besides the sulfur element, we can also see the carbon element, which is the surface coating. And then this is the TEM shows that both uncoated and coated nanosulfur shows a uniform spheroid shape, but with different particle size. The coated nanosulfur actually shows smaller particle size, maybe due to less aggregation. The XRD further demonstrate that the crystal of the sulfur material are all sulfur 8. Here we could see that they all show similar peaks and compared with the standard material. Besides that, we also test the hydrodynamic size and zeta potential of these materials. We could see that compared with the non-coated nanosulfur, the coated nanosulfur showed less smaller particle size, which is in line with the TEM image. And for the zeta potential, again, the coated nanosulfur has more negatively charged surface. Here is another group. The graph on the right side shows the time-dependent dissolution of sulfur. Although we could see some increase of the sulfur concentration, however, the concentration is very low. This is the sulfur dissolution from 200 ppm suspension. So I would say there's almost no dissolution of sulfur from all the sulfur materials, which is, I think, very reasonable. So after all this, we prepared the treated soil and prepared all the seedling and transferred the seedling to the pots. The pots are placed in the greenhouse here in CAES and in a randomized factory design. So I'd like to share with you some, not some, but many images that I took during the experiment. Here is the early growth stage of all the seedlings that we could see. There's no obvious impact of in any treatments. Here are the healthy plants compared with control, and the infested plants. So no symptom of the fusarium has been showed in the early stage. But after after the plant grows at the middle growth age, we could see some effect of the sulfur treatment. So I don't know if you could see clearly that on the right side is the control, and the left side is the Pristine nano sulfur at 200 ppm. We can see that we could see that an obvious it seems like significantly more healthy than the infested control at both 100 ppm and 200 ppm. Also, the coated nanosulfur did a great job in suppress the disease. And I would say even better, maybe even better than the non-coated nanosulfur. Here is the ionic sulfur. We can see that they're almost nearly the same with control, and in some cases, maybe even worse than control. The box sulfur also did, a good job in suppress the disease, but not as well as the nanosulfur and coated nanosulfur. Then at the end point, this trend are more significant. So when put every treatment together, we could clearly see the trend that in the healthy plants, the nanosulfur and coated nanosulfur, they increase the yield of the tomato plant, the biomass of the tomato plant as a nutritional effect. Still, there are some more pictures shows this trend. And then for the infested plants, we also see similar trend that the first line is the control, and then uncoated nanosulfur, coated nanosulfur, ionic sulfur. So here we could see that so compared with the middle growth stage, at the end point, there's nearly no growth in the control, but in the nanosulfur and coated nanosulfur treated plants, many of the seedlings are still seems like growing. To confirm this result, and also to study the mechanism of this result, we keep conducting a greenhouse experiment too. So with a similar design, we just repeat the experiment 1 and prepare the seedling and prepare the treated soil and do the transplanting. So in the end, we observed the same result with the greenhouse experiment 1. So here, these are all healthy plants, which shows that the coated nanosulfur and uncoated nanosulfur, they enhance the growth of the plants compared with the control and box sulfur. For the infested the treatments, same trend was observed in this photo, and this photo showed more clearly the trend. So based on this result, so those are the pictures for the data that I'd like to start with the disease progress. So from this one, we could see that the treatment with the coated nanosulfur and nanosulfur decrease the disease progress by more than 50% respectively compared with the control. Although the box sulfur also decreased the disease by like 30%, but it's not as good as the nano-sized sulfur. And the ionic sulfur had no impact on the disease progress. This graph shows the time-dependent effect of the disease progress, which sorry, I forgot to explain that the higher the number, the severe the disease is. So as the time goes by, the control shows the highest value, which means in the control treatment, the disease are more severe, are like the most severe. And on the other hand, for the nanosulfur and coated nanosulfur, as time goes by, that they remain at a lower level. That means they are less affected by they're less infected by the Fusarium disease. This graph shows the phytotopic data of the plants harvested at 4, 8, 16 days after transplanting to infested soil in the first greenhouse study. So there we could see a clearly time-dependent effect that there was no impact on the biomass as a function of treatment or disease at 4 days. However, starting from 8 day and 16 day, the disease reduced the leaf biomass compared with the healthy control, and the positive impact of nanosulfur were evident starting from 8 days. So the effects are most significant at 16 days. Sorry, the impact of bulk sulfur become non-significant, but the nano and coated nanosulfur increased the shoot biomass by more than 200% and 300% respectively, and also increased the root biomass by around 200% compared with the control. This graph shows also still the shoot weight and root weight in the second greenhouse study, which lasted for 65 days. Still, we see the same trend that with the previous one, that the nanosulfur and coated nanosulfur significantly increased the shoot weight and root weight compared with the control, and suppressed the Fusarium disease much better than the bulk sulfur. And the ionic sulfur almost are the same with the control. We also tested the photosynthetic parameters. So from here, that the LEF means the linear electron flow of in the leaves, which is positively related to the photosynthetic capability of the leaves. So we could see that in the nanosulfur and coated nanosulfur treated plants, higher LEF value are shown, and also we could see similar here the relatively chlorophyll and the leaf thickness, which are all show a positive effect by after treated with the nanosulfur and coated nanosulfur. The sulfur content and translocation ratio are analyzed by CAES, and from this graph that we could see disease had no impact on the root sulfur content at day 4 and day 8, but by harvest, I mean at day 16, the diseased root sulfur content was 40% greater than the healthy control. And all the sulfur treatments increased the root sulfur content at 16 days, although the nanoscaled sulfur treatment increased those levels more rapidly. And from the translocation rate, we can see that the box sulfur showed a different trend with the compared with the nano and coated nano sulfur, which may indicate a different uptake or translocate mechanism of the sulfur content. So we know that the biomass of the tomato plant had been significantly increased. We also want to know what about the nutritional element accumulation, which may related to the quality of the crops. So we analyzed all the concentration of all the nutritional element in the different tissues, different organs of the plants. And here are the raw data that I won't like discuss in detail. We summarized all the data in this heat map. So here that it shows all the nutrient element contained in the plant leaves, stem, and root tissues. I won't talk in detail, but from our result that we saw like different elements are differently affected by different treatments, and most the increase were shown in the nano and the nanoscaled sulfur treatments, however most of the element remains unaffected. To understand like why this change happened, we further analyze the extractable nutritional element content in the soil by extract the soil with different solvent. So this represent like the portion of bioavailable nutrient element that could be uptaken by the plant in the soil after the sulfur treatments. So from these results that we could see in the DTPA extraction, higher zinc content was found in the coated sulfur treated soil than the nanosulfur and box sulfur and the control. In addition, the coated sulfur result in higher soluble phosphorus content than the box in the same DTPA extract. However, such increment was not observed in the extracts by the DI water or the calcium chloride solution. And in general, the bioavailable nutrient element content in the soil was only like minimum affected by the treatments, which is good that we know that which is good because the sulfur treatment didn't like alter the nutrient distribution and availability in the soil, but they still successfully like enhance the growth of the plants. So we need to know like which mechanism caused this result. Besides the two greenhouse study, we further did a field experiment with the similar design, but we add one more treatment, which is the foliar applied coated nanosulfur, and still we obtain like a dramatically increase in the plant yield. So this number represents only the fruit of the tomato plants, and it was increased. I mean, for example, here doubled or tripled by the nano sulfur treatments. So I think the first problem, the research problem, like how to increase the yield of the tomato plant and how to suppress the disease from the fusarium, we have successfully like solve this question. And now still there's something remain that if you remember previously we are talking about the soil acidification by the application of sulfur based materials. So we kind of solve the first problem that to prevent this happen, we can decrease the amount of the sulfur that exposed into the soil by change the bulk sulfur to nanosulfur because it has higher efficiency. And then now the second question still remain that does the nanosulfur goes the same pathway as the bulk sulfur, which generate two hydrogen atom within the pathway and make the soil more acid. So to answer this question, we further design our study and mainly use two approach. One is the two-photon microscopy, and the other is RT-qPCR to understand the related pathway. So firstly, I would like to talk about the two-photon microscope. So compared with the the traditional confocal microscopy, it has much higher resolution and can penetrate deeper into to the biosamples, so which can give us a in vitro image of the material inside a living tissue. To achieve this, instead of generate one photon, it generates two photons at the same time. So which allow it to decrease the energy that needs to make this image and less damage the samples. So by this technique, we're lucky to have the results here that we found the fluorescent signal under this two-photon microscopy of the sulfur material are size dependent. Size sensitive. So here for example, the image E and K represent the uncoated nanosulfur without and with sonification. So before the sonification, we only saw like weak fluorescent signal, but after the sonification, we saw stronger, higher intensity of the fluorescent signal. This is because that after the sonification, the previously aggregated nanosulfur, they disperse better in the suspension, and more nano-sized sulfur start to appear. And to confirm this hypothesis, we further detect the bulk sulfur and the bulk sulfur in F and I. So no matter we sonicated the suspension or not, no fluorescent signal were observed because we thought it's because of the bulk size. And for the coated nanosulfur in H and N, before and after the sonication, we didn't see any fluorescent signal. This is because of the surface coating. The stearic acid, the surface coating, which contains mainly the carbon, which does not have any fluorescent signal. So after the surface coating, although it may remain nano-sized, we cannot detect it, which is still good. And also this ionic sulfur the sulfur salt, there's no fluorescent signal, which is reasonable because they are soluble. They're dissolved in the water, so no signal could be found. So based on this, that if we found any signal in our plant tissue, that means it's nano-sized uncoated sulfur. So this gonna help us to understand the mechanisms of the study. And as the results show that we found signal in the leaves of nanosulfur treated tomato plant, which demonstrate that the plant can uptake the nanosulfur from the root and then translocate the nanosulfur to the leaves within the nano size. Although we also found some signal in the leaves of bark sulfur, and here we hypothesis that this nano-sized sulfur may generated within the plant tissue. Also, in the roots, we also found similar signal in nanosulfur and coated nanosulfur treated plants, which demonstrate the uptake, the efficient uptake and delivery of the nanosulfur material into the plant tissues. To understand this this size-dependent result, that actually previous literature also reported similar findings, but with different types of nanoparticles, which is the zinc oxide. It shows that the fluorescence intensity is negatively related to the particle size. And once, if the particle reach the bulk size, like more than 300 nanometer, which is, for example, our bulk sulfur, they're gonna cause light scattering. So that's why we cannot detect any signal from those samples. The two-photon technology, microscopy not only can took photos from the sample, but can also make videos. So these are some picture that I took from the video that shows the in vivo 3D distribution of different nano sulfur materials inside the plant tissue at different depths. I won't talk detail here, but this helped us to understand a lot of the mechanism of the and the process of the uptake and translocation of this material inside the plant tissues. Another approach to understand the uptake of this elemental sulfur, elemental nanosulfur into the plant tissue is RT-qPCR. We analyzed the related gene impression. A gene impression related to the directly association of elemental sulfur, which is the sulfur 0, into the plant directly from the inorganic sulfur to the organic sulfur. And the results show that also approve our hypothesis that the nanosulfur and coated nanosulfur, they in the nanosulfur and coated nanosulfur treated plants, this process seemed to be significantly enhanced by those two bars. And it only appeared at 8 days, which demonstrate a narrow time window of this process. So based on the two-photon microscopy and this gene expression study, that we further confirm that the nano and coated nanosulfur are uptaken by the plants directly without go through the traditional sulfate pathway. And they can be translocated from the root to the leaves within the nano size and remain at like the sulfur zero chemical like the state. So right now we solve the question that what happens after those nanosulfur material are being uptaken into the plants. Now the next question for us is, what is the mechanism of the disease suppression? So again, we use two methods to study this mechanism. One is the RT-qPCR, and the other one is the metabolomics. So this is the diagram of our targeted gene that related to the sulfur bioassimilation inside the tomato plant tissues. So it's starting from here that the sulfate should be uptaken from the root into the plants, and then goes through this pathways and to form different metabolites that contain sulfur inside the plant tissues. So all this yellow gene that we designed by ourselves, which are we designed these primers to analyze the expression of these genes that within the bioassimilation pathway of the sulfur, to see like what happened and how the nanosulfur and coated nanosulfur and bulk sulfur differently affect this process. So here are all the data that we obtained from this RT-qPCR study. And to summarize, here is the heat map that and the DSPC network showing the results. We could see that the nanosulfur, bulk sulfur, and coated nanosulfur they affect like this process differently. So let me explain the DSPC network analysis a little bit. So this this bound indicate a relation, sorry, a relationship between the two genes, and the red color represent positively related, and the blue color means negatively represented. And more thicker the line is, that means more like related they are. This PCA score plot shows that at 8 days, like the nanosulfur and coated nanosulfur are more separately with the control group, which means the time is a very important factor that within this process. Here are more summary of how to understand this data, but because of the time that I will skip for now. I would like to further describing the results of our metabolomic studies. So we are able to detect all this different metabolites and like more than 100 different types of metabolites in the plant leaves, and the represented ones are I draw them into these graphs showing in different pathway. And there are mainly organic acid and also include some fatty acid, I mean, and others in here listed here. And this is the overall global metabolomic network based on the KEJJ global metabolomic network. And this PCA represent that both the time-dependent effect were seen in all the sulfur-based treatments. However, at 8 days, no significant difference was found between the control groups and all the other sulfur treatments. But at 16 day, the difference start to show, like the coated nanosulfur and uncoated nanosulfur are significantly different with the control group and the bulk sulfur group. This is in line with our previous data about the time-dependent effect. For the profile for the profile of the metabolomic pathway, we summarized here, and thus this is the comparison made between the nanosulfur and bulk sulfur at 200 ppm at 8 days after the transplanting. So the altered pathway has been listed here, and I won't describe in detail, but in the paper we like dig into, like we discussed all the pathway in detail and revealed the mechanism behind. So sorry about the time that I could only show you briefly the results. If you are interested, welcome to talk with me later or see our paper. And in the end, we did 16S RNA analysis about the distribution of the microbiome in the soil before and after the sulfur treatment. And thanks to Jackie and Blair, they did most of the work, and actually this graph are made by Blair. Thank you very much. This result shows that no significant difference of the number of bacteria are affected by all the sulfur treatments, which is also good. That that means our sulfur treatment didn't alter the biomicrobe distribution in the soil. For the summary, that in this study we observed the enhanced growth of the biomass and also the increased photosynthesis parameter of tomato plants. We saw that the sulfur treatment suppressed the disease of the Fusarium to the plants. To understand this result, we did the gene expression and in vivo and ICP element and the metabolomics. For the time-dependent effect, we use the ICP and the qPCR to understand this mechanism. Sorry about the time. I cannot read like one by one, but I should explain all of them in my previous slides. In the end, I want to thank all those people that help and support me a lot during this experiment. For example, at CAES, especially, I want to thank my PI, Dr. White and Dr. Emmert, and also Dr. Dimpa from DAC and all the DAC staffs, and also Stephen and like Jackie, who helped me a lot with the 16S microbes distribution, and also other cooperators in this study at UMass, at UTEP, and also in China, Zhao and Wang. Another people that I didn't put here is I want to thank the Yukon for the help of the LC mass analysis of the metabolomics. So thank you very much for listening, and please let me know any questions.
The question in the chat. Yeah. Right.
Yeah, I have a question for you. Yes. So I see that your ionic sulfur didn't have much effect. Effect was like the control. The ionic sulfur.
Yes.
Do you know why that is the case?
I'm thinking that probably about the concentration, and from the previous literature that we saw, the sulfur is not like the traditional nutrient. If you apply the sulfur like excessive sulfur to the plants, it may cause negative effect. This has been seen in many cases previously.
So did you try to link the effect on plant growth to loss of sulfur in the soil, to the amount of sulfur in the plant from the sulfate? But if you did that and you found all these pathways enhanced by ionic sulfur, by nanosulfur, and you're able to link that to the ionic sulfur and show a difference, your plant doesn't need the bulk sulfur, doesn't need the nanosulfur, it needs the ionic sulfur. That's what your plant needs to for its metabolism. If you're able to show a different pathway between the nano and ionic, more so than the bulk, I think that would make more impact. Because now we seem to have found a new pathway, but we really cannot prove that it's different from the ionic one. I don't see that I need data in the studies.
Right. Yes. This is because that we saw, because previously the ionic treatment didn't show much like positive effects. So in the second greenhouse study, which we want to study the mechanism, we didn't include the ionic sulfur.
Do you have those data?
For the ionic sulfur, we only have the data about the ICP, the biomass, but from actually from the translocation ratio of the sulfur obtained by the ICP. Here, the ICP OES, we saw that we actually saw a hint because for example here, the nanosulfur and coated nanosulfur, they showed a different trend compared. Oh, sorry. Sorry about that. Yes. Here so this is the root to stem translocation ratio, and this is the root to leaf translocation ratio. And the color maybe I'm wrong. Yes, you're right. So I'm sorry. So no ionic sulfur is included here, but we saw different trends when we compared the nanosulfur to nanosulfur, coated nanosulfur to the bulk. So we saw like this starting significant difference between the bulk sulfur and those nanoscale sulfur, which we saw that this may indicate may indicate a different uptake pathway. But yes, you're right. We didn't include ionic sulfur. Maybe we should look into that later on. Yes, please.
So you're talking a lot about you're using sulfur in the context of soil acidification, right? But you're doing a lot of plot experiments, and to have soil acidification, you need some leaching, right? Because you have to have the conversion to H2SO4, and then you have to have the SO4 to gel to be back to hydrogen, right? So I'm just wondering if, like, in the field experiments, do you see a difference in pH effects, or do you think you have to do or how are you doing the watering? Like, you're gonna have to have some, I'm just not sure you're gonna see acidification in a system like this because you don't have enough leaching.
Yes. Actually, I didn't show the data. We tested the pH of the soil like before the plant growth and after the plant growth, and we saw that based on the result, the nanosulfur and coated nanosulfur didn't decrease the pH of the soil, which is in line with our hypothesis. But to understand like more mechanism of that, were to confirm it is our hypothesis, we still need to design more study to demonstrate the pathway. But from right now, what we understand here, from right now, what we understand here is that in the traditional uptake pathway, the elemental sulfur need to firstly convert into the sulfate and then being uptake by plants. So during this process, it generates the hydrogen atom. But we demonstrate a new uptake pathway, which the plant can directly uptake the elemental sulfur without this process. So we saw that this may improve the situation and prevent the decrease of the pH of the soil. And I'm sorry that I just don't want to explain this, and actually I'm not quite understand your question. Could you ask again? Or
I'm just saying, like in a plot, you don't have a leaching of the SO4, right? So you're going to have microbes. Like, I don't know the plant. I think plants are still preferentially going to take up sulfur because it's like I didn't even know they could take up all of it. So they do it, but you're going to have microbes producing SO4, and then that has to leach out for acidification. So in a plot experiment for watering, you don't have the water flow through. You don't expect to see big effects on pH just because you're not leaching, because you're going to keep everything at a stable equilibrium. Right. So I'm just wondering if you're seeing something different in pH in the field trials, or if you could try watering and catching leachate or something to get that acidification, because I don't think you'll see that.
Yes, that's a good suggestion. Thank you. And I think I agree with you, because this experiment that we did is mainly for the mechanism study, is mainly in the greenhouse, and the soil that we're using is potting mix. So as you know that the potting mix does not contain a higher amount of sulfur. So we assume that, and also for the fertilizer we apply to the plant has an extremely low sulfur content. So we saw that the sulfur in the soil are all the sulfur material that we added into the soil. So they're mainly in the elemental sulfur. So in that case, in the greenhouse study, we I think we don't need to worry too much about the leaching, but in the field it's gonna be different. The field soil gonna contain a higher amount of sulfate, which I don't know how much, but should be higher than the potting mix. So you're right. So the field condition is different. Actually, we're now this is our first paper, and now we're continue with this work. We're gonna work with the soil, like the field experiment, and for that one. And thank you very much again for your help. We're gonna have further data of the soil microbe distribution, which could better understand what happened in the soil, which help us to better understand this mechanism.
There is a question, Joe, Interval asked you in the chat. Can you answer that?
Oh, sure. Thank you very much for asking the question. I don't know if I could share this question with all of you, but I can read. So you said that the addition of sulfur acidic make the soil more acid. So in your greenhouse experiment, did the soil pH depend on the form of sulfur that was added? And could that have had any effect on the growth health of the plant? And thank you for this question. I know many people are interested in the pH of the soil, and we're the same, where we care a lot about the pH. So we detected the pH of the soil right after the amendment of the sulfur material to the plant, before the plant grows and after the plant grows. So it actually are the results are different. I didn't show it here. So before the plant grows, we saw that after the amendment, immediately all the sulfur-based material, they decrease the pH of the soil. However, after we transplant the plants, the seedling to the soil, I mean, at the end of the experiment at harvest, we measured the pH again, and we didn't see significant change compared with the control. So so in our case, I cannot answer this question, like how the different pH like generated from the treatment affected the plants, because in our case, it doesn't happen because there's no significant like changes of the pH compared to the control at the end, which is good in our for our aim, that we don't want to see the soil acidification. So I don't know if I answered your question. If you'd like to ask more, please. Yes, please.
So I'm very interested in your metabolomics.
Yes. I think I'm starting here, and actually because the heat map, I cannot put everything here, so I only pick the representative metabolites, and for the other metabolites that we detected, I put it into this global metabolomic network, and then I can't say a major finding, because you know, this is a very big experiment. What we did is we try to include everything. So we have the factor of the treatment, I mean the different size, the nano size, the bulk size, the coated nano, and also we included the time factor. So we compared the changes of the metabolomic profile between 8 days and 18 days. So so there are many findings. This is a very interesting study, and we got a lot of like interesting results. That's why we thought this is a good, like interesting study. We should continue. So, but I could explain like the most significant one. For example, this is I said this is a time dependent effect, because at 8 days there I mean those groups are not separated. But at 16 days, we can see that the nanosulfur and coated nanosulfur started to change the metabolomic profile compared with the control and bulk. And then for the details, we analyzed the pathway. So, so for example, this one is the comparison between the nanosulfur and bulk sulfur. So once, if we saw like if we saw any pathway listed here, that means the nanosulfur has a significant different profile with compared with the bulk sulfur, and those metabolites that are mainly like play the most important role are listed in the second row. You can see that like the glutamine and many other metabolites that within different pathways, and they all have different function in the plant growth. So I cannot like give a very general conclusion here, because we discussed this in detail in the paper. It took us a lot of time to understand these results. And then this is a comparison between nanosulfur and coated nanosulfur. Then we saw different things, that there different pathways are changed and different metabolites are included. So so it really need to focus on like which cases and which metabolites that we're interested.
So, just follow up. So once we find the key points in metabolites, let's say, like by, is there a way to go back or have to run another experiment? Is there a way to analyze them? Really use the technology, but different set of analysis, like an easy way to go once you pinpoint the major ones, just maybe not the analysis to experiment.
I don't understand. Sorry, can you explain again? So you are asking how we understand this huge data set?
No, the data set is a fact.
Yeah.
But once you pinpoint the major ones, what would be the best way to further experiments? You just focus this major numbers. Like the best analysis.
I think there are two ways. One is you need to have your own hypothesis before the analysis. So that means you for us, we would like to know the sulfur contained metabolites. Like they're the most interesting for us. And or if you like observe significant changes with the phyto like the height of the, for example, the height of the plant has been significantly changed, so that you can check which pathway control the growth, like to control the height of the plant, and then you can like target, you can narrow down your interest, the metabolites. That's the one thing. If you don't have a specific hypothesis, there's another way to understand, to pick up the metabolites which is used the software. I mean, a software can help you to like give you an overall understanding and an overall profile of all the metabolites, and then you can figure out after that by yourself. But I think you need to work from both approach because the data set is, you're right, the data set is nightmare. It's a lot of data, a lot of information, and I think like one more day you can have more data and more results, conclusions. So you need to, yes, you need to be really focused on like what is the most important to your research. So I think that's why I think the metabolomics is a very powerful tool and analysis like way to help us to understand like the study from different areas, different aims, different approach, because this give you all the data that you can pick up by your own. Like a small part of the data can already solve a big research question. Sorry, I saw one more question, but